Welcome to the day 2 of the web series on health and diseases contemporary culture. This web series is organized by the Center of Excellence in Environment and Public Health, Ravenshaw University, in collaboration with National Academy of Sciences India, Indian Association for Neurosciences, and the Deputy Institute of Life Sciences, Bhubaneswar. It is also the part of the unique lecture series, Ravenshaw 150 Distinguished Lecture Series. This Ravenshaw 150 unique lecture series was organized by welcoming different distinguished personalities from the various corners of the world to celebrate the sesquicentennial year-old Ravenshaw University's achievements. So today's lecture on how virus affects human brain is a part of that Ravenshaw 150 distinguished lecture series. Today with us we have the Professor Pankaj Seth from National Brain Research Institute, a senior scientist from the Manesar Gurugram. He obtained his medical biochemistry in 1996 from Central Drug Research Institute, Lucknow, and was trained in neurovirologist during the postdoctoral training from 1986 to 2003 and Uniform Services University, Bethesda, USA. Dr. Seth worked at the National Institute of Health, Bethesda, USA, before returning to India to take up associate professor position at NBRC in 2003. Is actively pursuing various aspects of HIV-1 and Zika virus-induced neuropathogenesis with special emphasis on neuroglia biology using human neural stem cells as a model to developed by his lab. His research group was made landmark discovery that HIV-1 transactivating protein, the HIV-1C subtypes that is highly prevalent in Indian population is less neurotoxic to human neurons due to a natural mutation at the position number C30S31 in the uh, in a neurotoxic region of the TAT protein as compared to HIV-1B subtype. Dr. Seth's laboratory has successfully established a human fetal brain derived neural stem cell culture that is being used by several laboratories, including his own, to study various neurodegenerative diseases. His group is also engaged in research on HIV-1 and drugs to abuse comorbidity and role of microRNA on HIV-induced damage to the brain cells. His research has been supported by several funding agencies like National Institute of Health, Bethesda, USA, Department of Biotechnology, Department of Science and Technology, Government of India, and US-India Bilateral Cooperation Initiative of NIH and Indian Council, including some of the NIH USA. Dr. Seth served 2 terms as the member of Special Expert Group of Neuroscience, Chronic Disease Biology, constituted by the Department of Biotechnology, Government of India, till 2014, and a member again from 2016 to 2018. He has been recently nominated by Secretary Deputy to be a member of the new task force on chronic disease biology since September 2018. Dr. Seth is also a member of task force on stem cell research and regenerative medicine constituted by the Department of Biotechnology and also a member of Neuroscience Project Review Committee of Indian Council of Medical Research. He serves on various advisory boards and committees of national and international institutions and has because due to his immense contribution to the field of science, particularly virology and neurobiology, he has been elected as the fellow of the National Academy of Sciences India and fellow of Academy of Neurosciences India as well. With these words, I welcome Dr. Seth to deliver his today's talk, the Ravenshaw 150 lecture series, How Virus Affect Human Brain. Dr. Seth, please.
Thank you so much, Dr. Luna, for the kind introduction. I must add one more thing, that I have been very fortunate to be associated with Dr. Pathro right from the beginning, especially in the glial initiative, which was started at DBT. Dr. Pathro was very kind enough to include me in that group, which has done wonders for this country in terms of glial research. Dr. Luna, can you see my slides? Hello, can you see the slides? Can you see the slides? If someone can confirm if you can see the slides, I can move forward then.
Yes, sir. Yes, it's clearly visible, but it is in like tab mode. You can go by one by one.
Is it fine now?
That's good.
Okay. So the topic I've decided today to talk about is on how do viruses affect brain. And now, if that is a question, assuming that this webinar series also has some students included in it, a good researcher should have a question as to how research is to be done to prove that indeed viruses affect human brain. So research as such is kind of a puzzle, and I've used this slide which I used in my recent TED talk in 2020, where I tried to explain it to the school children also, that how research is done. Research is basically nothing like but a puzzle, which we don't do nowadays, but in my days we used to do put together pieces of puzzle to form a picture of a scenery or of a human being. Similarly, when we do research, we require patience, passion, and sustained funding support to put together all pieces of unknown ideas about this particular research. And then when we put all of this together, sometimes a PI puts it together, sometimes a collaboration puts it together, sometimes people from across the boundaries in other countries will, working in the same area, will bring together different pieces to form a picture. Having said that, if a researcher is there, the first question which should come to your mind is, how does viruses affect human brain? I started with saying that as a title, but the first question which should come, does the virus really reach the brain? If it does, what are the mechanisms, considering that there's a naturally selective blood-brain barrier which is provided by the nature? How does these viruses, or any pathogen for that matter, cross the blood-brain barrier, breach the blood-brain barrier, and then reach into the brain parenchyma to cause a disease? Once this virus, or any pathogen for that matter, reaches in the brain, what cell type does it attack? Because there are various types of cells in the brain. There are glial cells, there are neurons, there are oligodendrocytes, microglia. So what are the cell types which are being affected, and what decides that? Now, how does, whether HIV, Zika, or novel coronavirus, how do they decide which cell they have to go and attack, and then they go forward? So these are research questions which can actually cause, can open areas, and this is what I'm trying to ignite in young minds, how you should approach as a researcher. And if you understand that, let's say HIV goes and hides in the astrocytes, how does it affect the cell machinery when it gets into the astrocyte? Similarly, if the SARS coronavirus or the novel coronavirus gets into a cell, how does it affect the machinery? So my lab is actually working on two major viruses. And since I started my lab almost 15, 16 years ago, we started working with HIV, the causative agent of AIDS, and the other virus we got interested into was Zika virus. Now, the differences between these two viruses is that while this virus attacks the neural stem cells in the brain, attacks the glial cells in the brain, this gets into the brain by crossing the blood-brain barrier, and it cannot cross the placental barrier. So most of the effect of HIV are after the birth of an individual. However, Zika virus starts very early, and it starts its effect in utero when the and how does that happen? If the mother gets infected early in the first trimester, first 3 months of a pregnancy, it can affect the growth of the, especially the head size or the brain of the of this fetus. And this is possible because the virus crosses the placental barrier, which is not possible in HIV. But Zika virus crosses the placental barrier, and that is why it affects the fetus in utero, small size of the head. Now, let me try to explain you with the help of this animation how in a couple of minutes we will talk about and understand how HIV gets into the brain and what it does after it gets in. So once the virus gets, can be by various source of infection. It can be through heterosexual, men having sex with men, or contaminated blood. Once it gets into the blood, it goes also to the brain parenchyma, as you can see over here. The brain, where you see these endothelial cells which form the blood-brain barrier, you see an RBC, you see a monocyte, which is a blood cell. Virus also here, at least in my screen. I hope you guys can also see it. Smaller than this monocyte cannot go through this blood-brain barrier. However, this monocyte has a privilege to cross the blood-brain barrier, and this is because not it is infected, but in natural conditions and non-infected conditions, monocytes act like by crossing the blood-brain barrier. They are about to do so that they can go and check out in the brain parenchyma if everything is also differentiate into macrophages. So that is the normal function of monocytes. But the viruses are smart and they sneak into these monocytes, and then it barrier cannot detect that the monocyte is infected. It allows it, as you will see in rest of the animation. And this is what is called as Trojan horse hypothesis, by which the HIV virus gets into the brain. And you can see this monocyte infected, carrying the viral particles. It gets into the brain parenchyma, looks for a CD4 positive cell receptor, and it gets into the brain cell, injects its nucleic acid, and you just saw how a brain cell is being infected. Once it gets into the brain through the blood-brain barrier, as you saw in the animation, it can affect the neurons. Neurons are the functional unit of brain. Now, all neurons are always supported by various kind of glial cells, but astrocytes, which are one of the most abundant cell type of the brain. Now, interestingly, neurons never get infected by HIV, and that they do not have the CD4 receptor to which the GP120 of the protein of the virus can dock into and get into. So viruses are never in neuron cells, but they affect the neuron cells because they affect the neurons by two ways. Number one, they can shed these viral proteins, which is the GP120, which is highly neurotoxic. The virus also makes one other protein which is called as HIV TAT, and the TAT protein and the GP1 protein can cause neurotoxicity because they're toxic in nature for the neurons. So that is one way by which neurons can be affected without being infected by the virus. And the other thing is that these glial cells, which are supportive cells for neuron, get infected and they stop doing their functions. For that is they provide certain growth factors for the growth of the neurons, and they also take away the excess of glutamate from the synapses, because these astrocytes take away the glutamate and they recycle it so that the neurons do not die because of excess of glutamate, which is also called as glutamate toxicity. So these two important functions of providing growth factors and death of the neuron. So this is how astrocytes are, the neurons are being affected by the virus. One is the direct way. One is the indirect way, which is astrocytes are being affected. So irrespective of these two, either direct pathway or indirect pathway, neurons eventually die. And if the neurons are dying, they actually result in cognitive and motor compromise of the cognitive and motor functions, which is in AIDS patients, which is usually studied under the field called NeuroAIDS. And that is why we call our lab a NeuroAIDS lab. Under this field, we try to understand how HIV viral proteins affect the brain cells and their functions. And one of the challenges when we started this research, we as my lab as well as others in this field, was that because this virus is a human specific animal models. In other words, if you take this virus, put it in rats, mice, or cats, nothing will happen, but you accidentally inject yourself with the virus. Our was to find a good system which is of human origin, where you can understand how the pathogenesis of the virus occurs. And that is when we started working on brain cells. We started to have these aborted fetuses. After having all ethical clearances, we would collect these aborted fetuses. Early gestation was preferred because you can get best neural stem cells at the early stage of the fetal tissue, ideally 8 to 16 week. And this is what you will usually get, because after 22 weeks or 20 weeks, abortions are not legal, are not allowed, are not performed. So this was, you would get these fetal tissue, you do a lot of massaging. All this is published, or I'll be happy to share the protocols with you if anyone is interested. And at the end, what you will do, you will try to characterize whether the cells which you have are actually neural stem cells or neural precursor cells or not. So you use cell-specific markers which will define that they're stem cells. And the beauty of these stem cells is that they can be grown as neurospheres or as monolayer cultures, as you can see in this slide. So these neurospheres are basically typically only formed in brain neural stem cells, and they form this perfect sphere-like structure. And when we characterize them for stem cell markers, nestin is one of them. We found that they are nestin positive. They were also positive for SOX2, and they're highly proliferative, which we used also a cancer biomarker, which is Ki67. And they're Ki67 positive. So these things and many other tests which we did confirm that these human neural stem cells were indeed the cells which we were looking for. And as I said, they can also grow as monolayer or as suspension culture in neurospheres. Using this model system, we have published various papers in the field of neuroscience as well as NeuroAIDS, either from our lab or in collaboration, indicating that this model system is very robust. Now, how are these neurospheres formed? This is very interesting, is that it is not that all cells come together. It is basically one cell divides as the stem cells would, 2 into 4, 4 into 8, 16, 32, and so on. And they will form these sphere-like structures as is shown in this time-lapse microscopy. Now, this, if you measure the diameter of this and the time in a specified time, you count these cells when you started with one cell, and at the end of 48 or 72 hours, you get these number of cells. This is how you can also use as Neurosphere assay. Now, if these were neural stem cells, they should also differentiate into brain cells. So what we found was they do differentiate both in astrocytes as well as neurons, the two major cell type of the brain. And what we found was, as is shown over here, that the cells differentiate in green, which is labeled by green in neurons. And some of the cells are differentiating. That is why they're carrying both the nestin, which is the neural stem cell marker, or the early neuronal marker. And that is why they show yellow and orange. So these yellow and orange cells are under differentiation process. And the green ones are actually fully differentiated neurons. And as I said, these neural stem cells can actually be differentiated both into astrocytes or in neurons. So in this slide, you can see that you have neural stem cells, you have brain astrocytes, you have brain neurons. And then now we were ready to understand, to use these cell types into understanding how HIV causes damage. So the potential of this human neural stem cell model is immense, and we have used it in various ways to understand how HIV affects the stemness of neural stem cells, or how HIV affects the neurogenesis, effect on astrocytes, and how astrocyte-mediated neuronal damage occurs. So indirect pathway have also been studied using this model. And we published all these papers indicating that this model is very robust and very, very helpful in understanding how HIV virus affects human brain. And then we extended our research into Zika viral proteins, as well as now what we have planning is to use this model system also to understand how SARS-CoV-2 affects the neural precursor cells, astrocytes, and neurons. Because now there's mounting evidence which is available which suggests that this novel coronavirus or the COVID-19 patients are having neurological problems as well. So we are trying to understand how this virus, the new novel coronavirus, is affecting the human brain. Now, very quickly, without giving you too much of data, I will just walk you through as to how we understood how HIV is affecting neural stem cells. We started our work with a simple research question, because to find out if the TAT protein, one of the neurotoxic protein, as I told you, which kills the neurons, does it have any effect on human neural stem cells? So all we had to do was take this recombinant protein, put it in the neural stem cell cultures, and then try to see if the neural stem cell proliferation or differentiation is being affected. When we did that, the very first experiment we did was we took the neural stem cells and we added TAT, recombinant TAT, in the media so that to find out the effect. And what we found was very, very dramatic. What we found that if you have HIV TAT in the media, in controls, you will have the cyclin D1, which is which helps in the cell cycle, and the Ki67, which are pink or red cells, which you can see on your side. We found that the number of red cells goes down, and the green cells actually totally disappear. The green stains from the cell actually disappear, indicating that TAT is affecting the cell cycle regulation. So the proliferation was affected. What we also found was that the differentiation property of the neural stem cells make neurons and glia. When we tried to make new neurons out of them in the absence of TAT, we found in day 1, day 3, and day 5, you would see that the number of green cells are increasing as the day passes when you put neuronal differentiation media and these neural stem cells, as is indicated by the green stain. But in the presence of TAT in increasing concentration of 25, 50, and 100 nanogram per ml, you find that the number of green cells is very, very low as compared to on day 5 in non-TAT treated cells. Now, this clearly indicated that indeed the TAT affects not only the proliferation, as you saw in cyclin D1 staining in the previous slide, as well as in this slide that the neuron differentiation is also affected. We quantitated all this and published in this journal Neurobiology. This was a landmark paper because this opened new area of research where we had shown that HIV TAT protein, so how HIV is affecting the neural stem cell. So any chance of, when the HIV is damaging the brain of repair, which could happen by neural stem cells, is actually also blocked by the TAT protein or the viral protein. So moving ahead, then we had another question, because we wanted to dig into the mechanism as to how proliferation is affected. So one student in our lab started working on this for her thesis, and she found that we had a research question that if HIV TAT is actually causing quiescence or is slowing down the proliferation of neural stem cells, is it through something? There must be some molecules, some transcription factors, some genes which are regulating that. And we thought of TRIM32 because TRIM32 was shown in a Cell paper few years ago before we had published this paper, that TRIM32 actually regulates the neural stem cells of mouse. So what we did, since we had these human neural precursor cells and we had TAT protein, we asked the question if TRIM32 is regulating the quiescence of TAT on the neural stem cells. That was simple to see. So first what we did was, first of all, we looked at the nestin stain in the human brains of HIV individuals. So we went to we requested the NIMHANS brain bank, Professor Shankar at that time, and requested him if he could get some brain sections of AIDS patients. When we looked at these AIDS patient sections, we found that in HIV, the nestin, which was the neural stem cell marker, the brown stain of nestin was almost gone in these patient samples, brain sections, which indicated that the neural stem cell staining is low. And then we asked this question, if TRIM32 is important for this. So all we had to do was knock down the TRIM32 and then again put the TAT and see if the Ki67, which is the proliferation, is occurring. And indeed, this is exactly what we found. If you have TAT in the system, the Ki67 positivity cells are low. If you add TAT in the neural stem cells, the proliferation stops. If you take away the TRIM32 by using siRNA, the Ki67 cells come back, which means this is almost equivalent to what we are seeing in the scrambled siRNA, indicating that TRIM32 is modulating the quiescence or is stalling the proliferation of these neural stem cells. And that is what we published in this paper. We also narrowed it down to a microarray of 155, which was important for the HIV TAT mediated effect. We went ahead and did more experiments to look into how HIV is doing affecting the glial cells, because as I said, HIV kills the brain cells, particularly neurons, in two ways. Number one is by direct mechanism, as I showed you till now. And the other one was indirect mechanism, because it takes away the support and the goodies which come from the astrocytes to the neurons. So it attacks both the neuron directly without infecting it by viral proteins, and it also kills the support system of the neurons, which is basically the astrocytes. So in a review which we published in 2015 was very well cited, where we had shown that how astrocytes have a support neuronal base. And not only us, many people have published several reviews, and that is when the glial biology initiative was started by Professor Patro in India, where he showed the importance of glial cells and how glial cells contribute to neurodegeneration in case if these cells do not do their function properly. So taking lead from there, what we also did was we did a co-culture this time. So as I said, from the stem cells, we differentiated astrocytes and neurons, and then we started putting them together. And if you see closely over here, you will see that one neuron is actually surrounded by several A's, which is astrocytes. The flat cells, gray cells, are astrocytes. The spiny cell in between is the neuron. And if you look at the stained picture of the same co-culture, you would see that this green cell, which is basically a neuron, is trying to extend its processes and trying to make connections with the astrocytes. Now, why do neurons try to connect to these astrocytes? And why do neurons in culture grow better if they have astrocytic media or astrocytes in their vicinity? It is because at these yellow circles, this is where they form connections, and they're making these connections to get the support not only physically, but also a lot of the growth factors which they need for their growth. So if the neuron has to function optimally, it depends on the neighboring astrocytes. And that is why for one neuron, often there are 50 to 100 astrocytes in the vicinity. So what happens to this co-culture if you have TAT or any toxin in the environment? If you put TAT protein in the environment, what you would see, these neurons which were making connections start withdrawing from the astrocytes, and this is because the glial cells or the astrocytes become activated in the presence of viral proteins or the viruses. And then the neurons withdraw themselves from astrocytes, and that is because these astrocytes, which was actually providing support and good stuff to the neurons, actually starts producing certain cytokines and chemokines which are bad for the neurons. And then the neurons have this beaded appearance, and if you do not wash that away from this at this point, they eventually aid in the apoptosis of the neurons. So this is how astrocytes mediate neuronal damage. And Manju, one of the graduate students who joined the lab, showed that it is causing due to the activation of purinergic receptors on the astrocytes, and her work was actually featured on the Journal of Neurochemistry cover page. Then to understand more mechanism how purinergic receptors are activated in the astrocytes, Meher did another extra work, and she found out that the purinergic receptors are activated when astrocytes are activated, and astrocytes, among in addition to cytokine and chemokine, also produce a lot of ATP, and the ATP is actually coming from the bolted dependent anion channel 1, VDAC1. And this VDAC1 is regulated by a microRNA 320A. So I won't show you any data, but a summary slide where we have shown, or actually Meher has shown, that HIV TAT produces enhanced amount of ATP release from the astrocytes, which actually lead to the death of the neuron. And this is via the VDAC. Even the VDAC channel opens up, and the opening and closing of VDAC is being regulated by the microRNA 320A. And how do I know this 320A is important? Because if you use a mimic or inhibitor of this microRNA, you actually regulate the VDAC, and you can have the death or do not have the prevent the death because of the astrocyte ATP. So in this paper, which was published in Glia 2017, she had shown that HIV TAT is affecting glial cells, affecting the neuron damage. Now, very recently, we published another paper in Journal of Neuroinflammation, where again, this time Priyanka has shown that it is HIV virus and its viral protein TAT causes the astrogliosis, which is basically activation of astrocytes, which eventually leads to the death. And if you have this model in molecule, which you can actually read in this paper, I won't go into detail in the interest of time, is that it can help in preventing the TAT-mediated toxicity. This is the most recent paper from our lab. Now, very quickly, as I said, the lab is also working on another virus called a Zika virus. We work on both HIV and Zika viruses, and we use a variety of tools to understand how these two viruses or they affect the crosstalk between the neuron and glia. And that crosstalk is the same thing where neurons is dependent on astrocytes for its functions. So using these various techniques, lab is trying to understand various aspects of neuron-glia crosstalk and how these crosstalks are affected and how neurodegeneration is brought about. Now, HIV, after HIV with HIV, we also started working on Zika virus. And as I said, Zika virus affects the fetal development in the uterus. And when the kid is born, it is actually born with almost half the size of his head. So his head size is abnormal. And what is this indicating was that in uterus, the development is being affected. And the development affecting is because the growth of the brain cells is not happening. So we very quickly could understand perhaps why it was happening. And does it really happen? Yes, it's not only that cartoon which you saw in the earlier slide. These are the normal, these are the kids which were born to the mothers which were infected with the Zika virus. And you can see that the whole head size is small or their abnormal heads. These kids have very devastating outcome in their life. Some of them die during stillbirth. Others basically, if they continue to live, they have a very terrible life because they will never be able to cope up with their brain development later. Because all which has happened very early in their lives will have a long-lasting consequence on their development later on. And this was very well correlated with huge number of these cases of microcephaly as well as GBS, another syndrome, a neurological syndrome, with the infections which were reported or the outbreaks which were reported in 2016. And in every month passing at that time, you would see the more and more countries and territories were being affected, something what we are seeing as SARS now. And was it relevant for India? Yes, because India also had a Zika virus outbreak. Now, fortunately, we all developed herd immunity against Zika virus, but you never know if the virus mutates again, we can have the problem. And that is why it's important to understand how Zika virus causes microcephaly. So at that time, what was happening was there were a lot of paper, very big journals which were publishing that Zika virus is affecting the neural stem cells, it was affecting the human organoids, and so on, in animal model, in cell culture model. But nobody knew at that time which component of the virus is actually doing the damage to the progenitor cells or the neural stem cells. And that is why the growth of the brain does not happen. That is, at that time, we used different viral proteins of the Zika virus, both the structural protein as well as non-structural protein, and we transfected our neural progenitor cells with these viral protein to find out which part of the virus or which viral protein is having the maximum effect on the proliferative rate of these neural stem cells. And what we found was that the envelope protein which is enveloping the virus, or the E protein as it is called, of the Zika virus, is actually affecting the neural stem cell proliferation the most. And we use the Neurosphere assay, which I showed you. When we have E protein in the culture system by transfection, we have more of these smaller neurospheres, indicating these spheres did not grow as normal spheres, which were much bigger in the same time. When we started with the same number of cells, after 24, 48, and 72 hours, what we found was the cells with E protein had smaller spheres, and the ones without E protein had a bigger sphere, indicating that the proliferative ability was affected by the E protein. Differentiate into neurons, you would find that if you have vector control where there was no E protein, and with E protein, you can see that the new cells which are migrating neurons are not only migrating to longer distance from their origin, as compared to an E protein, this was very small, and the branching also was very beautiful in the controls, but not in E protein. Now, this was very useful, and when we sent this paper for publication in Cell Death and Differentiation, the reviewers asked us, "You're showing everything in Petri dish. Why don't you do it in an animal model?" And that's when we tried to collaborate with Dr. Jonaki Sen at IIT Kanpur. And what she did was she took the C protein and put them in uterus of the animals, rodents, when they were at the early stage of growth in the uterus. And these were put by electroporation. The E protein was electroporated into the developing fetal tissue, mouse pups in the uterus of these animals. And at 15.5, these were harvested, and we found that E protein was indeed there. And you could see that look at the width of this brain layer, one of the layers, and the red cells is SOX2. SOX2 is a marker again of neural stem cells. So if you compare red cells from this side in empty vector or in presence of E protein, you can easily see the number of red cells is low. So what is this? The stemness is gone because of the presence of the E protein. We also looked at the Ki67, which is the marker for neural proliferation, and you both at the end is the you look at the again Ki67 marker, which will tell you how many cells are proliferating. On this side, E protein is very few cells are proliferating, and the E is green cells are only showing that the cells are transfected. So these two data slides showed you very clearly that indeed the E protein is altering the property of human neural stem cells, and they are not allowing this Zika viral protein, envelope protein, is not allowing the virus neural stem cells to proliferate, and that is why perhaps the normal growth of the infant is actually inhibited because the cells do not divide and first trimester, first 3 months of their growth in the uterus of the mom. Now, coming very quickly in next 5 minutes, I will try to viruses. I am sure with all the popular media and as well as intellectual reading, you all smell been included by WHO as well as ICMR as one of the symptoms for in COVID patients, and we are seeing that later in MERS in the Arab area, we found that it was spread through camel. In been attributed to the antelopes as well as the penguins and the snakes, and all of these are said to be coming from a bat reservoir. Now, that's also interesting why bat have all these viruses, whether it is SARS, MERS, COVID, and maybe God knows in how many. But if you see this duration between SARS tenure between MERS and COVID, it is 7 years, and we don't know how when next pandemic is going to come. All is coming through the bat, but the bats have to live. This is also an interesting area to understand how viruses find bat as reservoirs, and nothing happens to the bat, but it causes devastation to the bodies. So let's try to understand very quickly how the SARS gets into the cells. It uses the ACE2 receptor and TMP-RSS2, which basically cleaves the ACE spike protein, and you finally have the cell like any other virus support. It goes into various tissues and the virus is budded off. Okay. Now, how does it get into the brain? We all know that these COVID patients are having multiple problems. They are having stroke, they are having intracellular hemorrhages, they are having thrombosis, they are having, as I said, confusion, headache, and also continue to have these problems, and they're not normal. You know, you would have heard a lot of patients who have recovered also continue to show problems of their confusion, or they're not able to think properly, remember properly. So this is what is indicating that this virus, novel coronavirus, is also affecting the brain. So how does it get into the brain is very important question. So we recently published a review in ACS Neuroscience in June, and at that time itself, before we had started to, we suggested that perhaps it is getting in through the cribriform plate, one of the areas which has limited blood-brain barrier also, through the olfactory neuron, all through the olfactory epithelial cells. And these cells, since they have direct connection into the olfactory, this is perhaps from the nasal, from the olfaction or the nasal area, where this is where, these are being collected. So the nasal swabs are taken for the same reason, because the virus is there in the mucous membrane of the nasal passage. And since these olfactory neurons have connections from here, from the nasal passage to the brain, that is why perhaps this is one of the route by which the virus is getting in. That is why your sense of smell goes away, because you damage these neurons. Interestingly, these cells, when the patient recovers, this sense of smell. So perhaps either there's a good rich of olfactory epithelial cells over here which make the new neurons, or some of the neurons basically come back. Interestingly also, because they are cytokine storm and the virus is present in the bloodstream, and through the blood-brain barrier, it can either go through, like Trojan horse hypothesis, something which I showed you in HIV case, it is not very clear, or when there's a cytokine storm, the blood-brain barrier tight connections actually loosen up and the virus gets into the glial cells, and that is why glial cells again finally play a role in spreading the virus or having the bad effects of the virus into the brain. All this is research where people are trying to do, and we have started our research. Have seen that diarrhea is also another symptom which has been added in COVID patients, and that is when the enteric lining or the enteric. Now, gut has a brain connection. We all know that the brain and gut are very closely connected, and that is through a lot of. Also, the vagus nerve connection has also been connected here. So, virus can go through olfaction, through blood-brain barrier, through gut. Eventually, it ends up into the brain. And all these are area of research which are new, and people are trying to understand how it does, and perhaps we will know very soon. Now, various papers have been published in last 6 months, actually huge number. It is very difficult to actually catch up with the literature of SARS. So what we have done in our lab, we have divided ourselves into small groups, and we, each one of us are trying to follow one field, like somebody is looking at astrocytes, somebody is looking at neurons, and so on. So in my lab, we are trying to understand how various ways by which the virus gets into the brain, and once it gets into the brain, what it does. In one of these papers, which is published in Journal Neuroinflammation, a very good journal, where they've shown that whatever happens in the systemic system through the cytokine storm, it eventually leads through the blood-brain barrier into the the inflammatory cells get into the brain, and that is what is causing inflammation. And then if there is inflammation, it causes damage. Now, there's also proof now, again, in some postmodern studies, that which is unfortunately not happening in India right now, but studies in Germany and China are coming out, which is basically showing that in the blood-brain barrier, there is leakage of the blood-brain barrier, and the virus presence has been shown by electron microscopy. And very quickly, how people are understanding what is SARS-CoV-2 doing in the brain is by using the induced pluripotent cells. So most of you would have studied or know what are human iPSC cells. Basically, you can take cell of your body and you can take them back into their in history. Basically, take them back and make them stem cells. Neurons, astrocytes, microglia, oligodendrocytes, and whatever cell you want of the brain. And people are also making brain organoids or mid-brain organoid. And then you can put the organoids, which are basically mini brain, on the brain. Now, some of the studies which are now clearly shown, symptoms COVID patients are showing, now we have started to understand what is happening. And what we know from literature, which is very recent, is that if you have astrocytes or microglia cells on neurons, and if you put the viruses, you can see the viruses capable of infecting these brain cells. This is first data which is available. In the brain spheres, which is basically a collection of cells which are similar to brain, which is of human beings, not animal models, there also they have now evidence to show that the SARS-CoV-2 viral proteins are present in neurons, which are shown in green over here, or also in some glial cells. Now in this slide, we are showing only neurons. It has also been shown that it is affecting some of the stem cells. This is all information which is available till now. Now, in the interest of Professor Patru, sir, microglia cells have been found to be affected. What they did in one of the studies, they have shown that if you have mice expressing human ACE2 receptor in, and you put the SARS-CoV-2 infection into them, at fourth day, these microglia have morphology, but on seventh day they move from into ramified. The microglia are activated, and you have the activation of microglia at 7-day post-infection of the SARS-CoV-2. So microglial involvement is there, sir. And there's of course some evidence also biochemical that you have GFAP and NFL, which are two markers, and they're showing up in the plasma also. When you have to study brain, best is CSF, but even if it is showing in plasma, that means there's a lot of damage happening into the brain. And this is what which came out in June this year, is that SARS-CoV-2 has effect on the neuron, loss of sense or smell and taste, causes brain lesion, encephalitis and meningitis, can cause aneurysm, stroke, and brain ischemia. And this was very interesting. We had actually submitted this in April, very, very early, when we were just about to understand this disease, and we had predicted that this may be happening. And incidentally, later on, what we found was that indeed this is now very prevalent, and stroke, clotting of lungs, different is happening in COVID patients, and people don't know what is happening, why it is happening. So this review is very, very informative. I would encourage most of you. It is the PDF of this is actually available on NBRC website if you go under COVID research, or you can write to me. That is a better way. I will send you the PDF. So we are planning to use the same model system which I showed you earlier for HIV and Zika. We have all the plasmids or the proteins of this SARS-CoV-2, and we are going to study them same way which we did using that model. And if that's my last slide, is the summary. So indeed, the viruses affect the brain. There's no question. So we have in the last 15 years, we have invested and contributed very, very significantly to the field, where we had shown how HIV affects the cognitive and motor a decline in AIDS patients. And we have now very well understood how astrocytes are affected and how astrocytes away, and the crosstalk between astrocytes and neuron is affected, and that is why viral proteins of HIV cause neuronal damage. Significant contribution of NBRC to this field. Secondly, we had shown that the Zika viral E protein or the envelope protein causes microcephaly, and that is because it damages the properties of neural stem cells. We have also looked at certain microRNAs, and the work is in progress, and we are one by one we have out which microRNA. So we, as basic scientists, leave it to the rest of the people to take leads from there and target each of these markers, perhaps prevent the microcephaly or bad effect of this virus, or Zika virus E protein. And there's growing evidence, as I said, of SARS-CoV-2 including are lacking. Mostly it is by iPSC brains or brain organoid cells, detailed mechanism, and we have already started working on it. So at the end, I will just make a final statement saying that viruses do affect human brain and by various ways, and understand the mechanisms of neurodegeneration, because unless you know how viruses or their viral proteins are bringing about damage to the cells, not make any therapies. Had we known, had we studied the SARS coronaviruses much in detail, today we would have a situation in SARS-CoV-2. And that is why when emerging viruses come, one should not give them give away research on them if the death rate goes down, because it is important to understand the biology or how pathogenesis occurs with this virus. With that, I will thank you all for your patience. I hope I still have some audience left. These virtual lectures are very bad because you don't see if anybody is there on the other side. So with that, I'll stop sharing the slides so that we can take any questions if they are allowed during the session. Thank you so much.
Okay. Thank you so much, Professor Pankaj, for your illuminating talk about how welcome, sir. How virus affects human brain. Virus infection from HIV, Zika, and COVID can affect brain through pathogenesis, neuroglial corrections, and most importantly, they can alter the microenvironment around and the ability of the neuron. That means there is a lesser chance to replenish these neurons after viral infection once these neurons are dead. So it was really wonderful to know, your discussion has highlighted everything from how virus enter or challenges the leaks the blood-brain barrier, enters into the nervous system, and there it starts affecting neuron directly as well as indirectly via using machinery of astrocytes. So it was really wonderful, and also you have your suggestion to use organoids or the culture cells to study this virus is a good approach. So I hope the audience and the researchers will take your advice, and I'm sure that your current work is going to be a good resource for the ongoing COVID research in India, and we hope that we will have very fruitful outcome out of it. So I would like to say that it is a very good time to have neurovirus talking when we are all so thank you for presenting your work, and I'm sure that the audience will be benefited with this your presentation and talk. And I would like to take some of the questions from the audience. So I have one question from Rajan Tiwari from BHU. He's saying that olfactory neurons are dying or supporting cells. So he means possibly that if the neurons are dying directly or the supporting cells, because if neurons are dying, it is hard to get back the smell, but COVID patients get their smell within 10 to 14 days. Yeah. So, sorry, your address to this question, sir.
Nice question. And we are trying to understand as to what is happening. So perhaps the supporting cells are dying first. And if it continues on high viral load, some of the patients take longer to get their sense of smell back. And you are right, if the neurons die because they do not divide, it is very hard to differentiate, replenish those neurons by division. But since the olfactory mucosa or the olfactory, there's good support or a good supply of olfactory epithelial cells. So one of the source of neural stem cells, also a lot of people have done, especially Dr. Ashok Agarwal of ITRC, and then later on, I think Rajneesh Chaturvedi is following it up at IITR Lucknow, where they have found that the olfactory epithelial cells or the sheath cells actually have the ability of having these neural stem cells. So perhaps it is possible that maybe some of these cells are being replenished, because it's not only SARS-CoV-2, other viruses also take away the sense of smell. Even in flu, you will see that sometimes you don't able to smell, but the flu recovery of loss of sense of smell is very fast, but it is taking longer. And some of the patients actually have lost some of the smell sense quite for longer time, and that is worrisome. So we are just about to understand how this is happening. I do not have a full answer for this question, for the simple reason we have not done research on this area as yet.
Exactly. So we have to wait for Dr. Rajan. We have to wait for more research, ongoing research, and we hope that we have more answer about this. So we have another question from Sahil. Is there any possibility that elder may also suffer from neurogenetic symptom and cardiac dysfunction? Okay, so basically, I don't know what he is asking in relation to virus, but yeah, so he wants to know if the elderly, how the impact of this virus infection to the elderly population is. So if you can address this question.
So elderly anyway have a problem of the pool of neural stem cells. We know that when the kid is born, and as we grow old, the number of stem cells actually come down. So Rakesh may have more stem cells than I do, and the gentleman who asked the question may have more stem cells than Rakesh does. And as we grow old, the pool of these neural stem cells go down. So any recovery for that matter will be much slower and more limited in cases of elderly. That is for sure. Okay. And that is true for any disease for that matter. And that is why perhaps the Alzheimer's, as we say, the damage to the neurons, it starts to occur, and it occurs more in the elderly patients. Okay. Because with age, the support system and the ability for the neural stem cells to proliferate and differentiate and replenish is very, very limited. It is there, adult neurogenesis is there, but it goes down. The rate goes down, the amounts go down, and the overall outcomes are that's why poor.
Wonderful, sir. So I have another question from Dr. Dhananjay Shoren from Ravenshaw Department, and he's asking, could you please suggest some cure towards NeuroAIDS? Possible cure for NeuroAIDS.
Oh, for NeuroAIDS.
Yeah, NeuroAIDS. Yeah.
So NeuroAIDS, one thing is very good which has happened for the AIDS patients, and that is the development of the anti-HIV drugs. Okay, with formation of these new drugs, which are very, very successful, and that is why if you look in the world, the problem of AIDS, which was usually used to be a death warrant, and I hope that becomes true for SARS also around the world in times to come. That initially, if you have AIDS, your death was pakka. Now, with these antiretroviral therapy, death is not happening. And how death is not happening is, we need to understand. These anti-HIV drugs actually take away all the virus which is present in the blood by bringing down the division or proliferation rate of the viral particles in the blood. Now, if the overall pool of the blood virus, HIV, goes down in the blood, then as I showed you in the animation, what that monocyte was carrying from one by one into the brain also stops. Because there's no viral load in the blood, and that is why damage to the brain goes away. And that is what we are happening to see, that after anti-HIV therapy has come in, the problem of NeuroAIDS has also gone down. So initially there was a lot of permanent damage in these HIV individual AIDS patients. But now the degree of dementia or degree of loss of memory, degree of cognitive and motor decline in AIDS patients has also come down. So we don't have a cure as yet, but anti-HIV drugs have helped in reducing the severity of NeuroAIDS. So when it will be cured is because now why there's a difference between that people are living normal but not mentally normal. This is because these anti-HIV drugs clear the virus from the blood, but the blood-brain barrier and tight junction in the brain, they are unable to cross the blood-brain barrier and get into the brain to clear the virus from the brain. So virus from the blood is clean, but from the brain is not that good enough. And that is why those symptoms of NeuroAIDS have gone, but they're not over. It will happen only when if we can eradicate the virus which is there in the brain. If we can clean with this virus, then perhaps the problem of NeuroAIDS may be limited, because it is there, and we know also now by research that all the virus in the brain is staying in the astrocytes. We can somehow flush them out from the astrocytes, then the problem of NeuroAIDS will also go away. That is what NIH is trying to do. They have invested huge amounts, and they have a program called eradication of the virus from CNS. And when that happens, then I'll have to do something else. I'll have to close down my lab with NeuroAIDS.
We don't hope so. Sir, I was getting interested about knowing, like you discussed about the HIV virus also modulating or influencing dementia. So I was interested. Have you ever tried the interaction between the STAT protein and the tau protein or the senile plaque during?
Yeah.
So
HIV causes early dementia or more severity of dementia. That has been worked out. And what we found was there's one enzyme called as neprilysin.
Okay.
Neprilysin achieves these things, and that actually plays around with neprilysin, and that is why deposition of tau and tauopathy is more in AIDS patients.
Okay,
one
thank you so much.
Any glutamate toxicity? There's one question.
So glutamate toxicity, I don't know. It is so much relevant to the talk. So there is one question: how does this storm trigger the SARS-CoV-2 contribution to the neurological? So we have limitation to take like questions. We will mail you the remaining questions, sir. Some which are more pertinent and interesting. So we will mail you all those questions. So with this, I really thank you so much, sir, for giving wonderful, informative talk. And I'm sure that audience would have really got a lot of information about this current ongoing SARS research. And with these words, I now welcome or invite our Honorable Vice Chancellor, Isan sir, to his presidential address. So, Isan sir, please.
Namaskar. A very interesting lecture indeed, Pankaj. I'm so happy about it. I would, Rakesh, you left the last question, which was asked by Dr. Prahlad Seth. He wants to know, will entry of virus through nasal route will be more damaging than through the inhalation route? That's a very interesting question. Pankaj, are you able to listen?
So, yeah, that's one of the tough questions, and my dad always asked us difficult questions. So, let me try to answer that. And, sir, inhalation that is why, we are worried about this droplet infections. Being a droplet infection, that is why we are wearing the mask. And the mask is being worn because we do not want this virus to actually come in any contact of either the nasal through inhalation or through the other droplet which are. So even if you touch your eyes, now one would wonder what is the effect, connection between eyes and the lungs or eyes and the nose. Now I will tell you this. Actually, I learned from Professor Tandon, who's my mentor and also mentor of Professor Patro, our president of NBRC, Professor P.N. Tandon. And sir told me that, when you touch your contaminated hand or finger with your eyes, actually what will happen is that the virus will get through the lacrimal gland connection into your nasal thing. If you put any eye drop, you can smell it actually in your nose. So there's a connection between eyes and nose and inhalation. So this is how nasal connection and inhalation, I think they are both going to go through the same route. And no matter if it gets into the nasal passage or it gets into the lung, it is going to end up causing the infections. Incidentally, lungs have the lowest amount of ACE2 receptors, it is said, but they still cause damage because it goes into other cells and clogs the lung parenchyma. So I don't know if I answered this question correctly. I will have a discussion with him later on this to understand more about it.
It's a it was very nice, very lucid way you really described how the viruses enter into the brain and how they affect the functioning of the brain, and more specifically how HIV causes cognitive deficits, or the Zika virus really stops the correct way of development of a brain. And then into the new COVID virus, how it is affecting the brain function. And more importantly for everyone, I must say that we have been struggling into a very new area in past couple of decades, I'll say, maybe 20, 25 years, when people started thinking that glial cells are not really the glue cells. They've got a lot more to do. And now we understand if you remove the glial cells from the brain will stop functioning. So everybody was thinking the neuron does everything. Now we understand glia does much more than the neurons what they do. Possibly the neurons only conduct and communicate, and the rest of it is regulated by the glial cells. That is what is going to come up very soon. And in the entire lecture, you would have come across that every time he talks about the astrocytes, about the microglia, about of course he didn't use that term, but what he talked all the way is the neuroimmunology. That is what it is, very very important in way of understanding. And I'm very fortunate that I had an opportunity of working with Pankaj. We worked in a group of about 13 neurobiologists, leading neurobiologists of this country, trying to enter into a very new venture, glial cell initiative in the country. This has been a very interesting lecture, and we also understand through our work that multiple stresses, particularly the viral infection during the perinatal period, that means during pregnancy or immediately after birth, also leads into a lot of changes, which almost looks like there's accelerated brain aging, or towards the aging changes, or changes of the disorders which really come with age, like Alzheimer disease, Parkinson's, or ALS, or some of the developmental disorders like autism. So it's been a very wonderful session, Pankaj. Thank you very much for agreeing to deliver a lecture to our students, faculty, and the scholars. And I see many scientists from outside. Dr. Sukla Prasad is also visible there because he put a note for you. He appreciated your talk writing. This was indeed a wonderful and highly informative virtual talk, he says. So there are many other people. I'm not able to really pick up all the names. And this has been a very successful lecture, Pankaj. There have been more than 340 people listening to this lecture through YouTube. Thank you very much. And I'm indeed grateful to you for accepting this. And to the participants, I must say that we are going to have many more lectures like this. Please be with us. I leave it to Luna now for the next proceedings.
Thank you, Shar, for your nice summing up of the lecture. And we are really indebted to Dr. Pankaj for this illuminating talk on the glial cell biology and the brain function and the effect of viruses on the brain. So thank you, Dr. Pankaj, again. So today we have come to the end of the second day, and this series, as Professor Patra has already mentioned, will continue till 21st of this month, every day at 4 o'clock. But there are three diversions, because we have people from Far East and the USA. So those two dates will be different. But tomorrow again we are meeting at 4 o'clock. Thank you for being with us. Tomorrow's lecture is by Dr. Sunil Raghav from Institute of Life Sciences. Institute of Life Sciences. So therefore, I request you all to be tomorrow with us. So it's a continuation of this SARS-CoV-2 biology lecture, and we will meet you again tomorrow at 4 o'clock. Thank you. Thank you, everybody.